Thanks, Wei for the great introduction. So as you said, I'm going to talk about our paper that just recently came out in Circulation looking at right ventricular sarcomere contractile dysfunction in heart failure with PH or pulmonary hypertension. So, given this is the translational section, I thought I'd start off with the clinical problem. And our clinical problem is specifically right heart failure. So half of all patients with heart failure and reduced ejection fraction develop pulmonary hypertension. And right heart failure in this situation, once present, worsens outcomes and prevents bridging to advanced therapies. So shown here is a Kaplan-Meier survival curve stratified by one hemodynamic index of RV function, known as pulmonary artery pulsatility index, or PAPI. The formula for PAPI is shown on the left, and what you can see is it's the ratio of PA pulse pressure, which is the difference between PA systolic and PA diastolic pressure, and right atrial pressure. And patients with low PAPI have RV dysfunction. And as shown on the right in the Kaplan-Meier survival curve, you see that patients with low PAPI have only 25, maybe 30% survival at 180 days. Moreover, we don't really have any good marker of RV function that clearly delineates RV failure from impending RV failure, which leads to the possibility that RV function is much more heterogeneous than what our clinical data is able to quantify. And so here we're testing the hypothesis that underlying RV cellular dysfunction is present in RV failure from half-breath PH with varying severity, but is not captured by the standard clinical indices of RV function. And so to explore this question, we looked at explanted hearts, and specifically we looked at right ventricular septal tissue from 23 half-breath PH patients and 9 non-failing controls, which are age and sex matched as shown below. And you can see that these patients have uniformly depressed LV function. You can see the ejection fraction is 23%, and it's very, very low in all patients. And the LVs are quite dilated on echocardiography. However, when we take the same patients and look at their RVs, at least with right heart catheterization, we see that RV function is quite heterogeneous. So shown here is PAPI, which is that same index I talked about on the previous slide. And you can see that the coefficient of variation is 144%, which means that there are some patients with normal RV function and some patients with markedly reduced RV function. And so we asked the question, is it possible that the patients with reduced RV function have depressed RV contractile function at the cardiomyocyte level? And so what we did is we isolated skinned right ventricular cardiomyocytes, one of which is shown here, and we acquired standard contractile indices of cardiomyocyte function, specifically the isometric tension-calcium relationship, the power-load relationship, or the Sarnoff relationship, and then the link tension or Frank-Starling relationship, also known as link-dependent activation. And we acquired these from all myocytes from all patients, and we shoved all the data into a machine learning model. And we then asked, okay, what does the machine learning model tell us? And what do the clinical differences look like between these two myocyte contractile groups? So the machine learning model gave us two groups, A and B, and we then just compared ad hoc statistically, what are the differences in clinical indices? And we found two clinical groups. First, we call RV decompensated and red, and these patients have elevations in right atrial to pulmonary capillary wedge ratio, which is indicative of RV failure, as well as depressed PAPI, compared to the blue group or RV compensated myocytes. And so this is quite interesting because now we're able to actually predict clinical RV function simply from cardiomyocyte contractile indices in skin cells. But the question is, which of our contractile indices or which combination of contractile indices is really underlying what we can predict? And so what we did was we decided to look at each of the relationships we measured. And shown from left to right is the isometric tension calcium relationship, power load, and link tension relationship, the latter being an active activation during 3.8 micromolar calcium activation. And you can see it for black controls or non-failing hearts, and then in red for half-ref pH with RV dysfunction. And you can clearly see that in all these relationships, maximum tension is down, power is down, and the slope of the link-dependent link tension relationship or systolic stiffness is reduced. But what do the RV compensated myocytes look like? So what we can see on the left is that the actual isometric tension, at least at these systolic levels of calcium, is actually pretty similar to controls. However, the power load relationship is quite reduced, and you can see that the maximum power is actually quite reduced, and whatever changes we have on the x-axis, we can attribute to the isometric tension relationship on the right. And we see a similar phenomenon in the link tension relationship, where we see that the slope is reduced, it's just that the intercept is not consistent with the isometric relationship on the left side. So what this tells us is that actually the only index that we're able to predict using clinical from clinical measures is the isometric tension calcium relationship. And in fact, we see that the power and the systolic thickness are both really, really depressed, but the clinical indices are not. And this is quite surprising to us, because we would assume that our clinical indices are quite good at least capturing some of these underlying behaviors. To kind of bring the point home, you can see a table comparing each of the different measurements between the two groups. And again, the only significantly different one was isometric tension. In this case, what we did was we decided to all right. Okay. And when we look then at the mechanics in these patients, we again see the exact same behavior, that the isometric tension calcium relationship is pretty normal for the RV compensated myocytes compared to the decompensated myocytes, whereas both the power load and the tension length relationships are equally depressed, similar to what we saw before. And so this relationship is bidirectional, and we've effectively established that this is a significant clinical predictor. But again, we really are interested in this. Why is it that we cannot predict power and length tension relationship in patients without RV failure? And so what we decided to do then was look kind of at the specific molecular mechanisms behind each of these deficits. And we're going to go ahead and start with the one that was easiest to target, which is the power load relationship. So I moved it over to the left of the slide right here. And we decided to look at myosin isoform ratios because it's been well established that shifts in myosin isoform can actually, from an alpha myosin isoform to a beta myosin isoform, can actually contribute to depressed power. And so we did bulk RNA-seq and we quantified the ratio of gene expression and beta myosin, which is MYH7, that's the gene name, and then to alpha myosin, which is MYH6. And you can see that in both the RV compensated and decompensated patients, there's a clear alpha to beta myosin isoform switch consistent with the equally depressed power in both groups. We then looked at targeted sarcomere proteins just to confirm that these weren't really different, and that's shown on the heat map for a whole host of them right here. But it's safe to say that they're all not different from each other. So really, the only thing that the RNA-seq really told us was that, yeah, a myosin isoform shift is present, and nothing else is really perturbed. But RNA-seq gives you a lot of information besides just simply, sarcomere gene expression. And so we decided to look at everything, which we can do. So here's a volcano plot looking at HFrEF PH patients compared to non-failing control patients. On the y-axis, you see the log 10 of the p adjusted, and on the x-axis, you see a log 2 fold change, and our cutoffs for significance are shown by these dashed lines. And we found 933 differentially expressed genes between HFrEF and non-failing controls. And again, this is nothing new because people have shown, several large changes in gene transcription overall in heart failure. And what we found when we looked at pathway analysis was that upregulations in striated muscle differentiation, sarcomeroskeletal pathways, and downregulations in metabolic pathways, all consistent with what's been published previously. So this is kind of a sanity check for us that the RNA-seq data makes sense. But then what we did was we decided to look to see, do the two HFrEF PH subgroups have any gene differences? And we literally saw nothing. There were 0 genes actually different between these two subgroups. And this is actually really, really surprising to us that we were expecting to see, something, but the fact that there's nothing is quite interesting. And it really suggests that the majority of the contractile deficits we've observed must be due to post-translational mechanisms, which RNA-seq is not able to quantify. And so, at this point, we decided to just look at the sarcomeres. And we were kind of scratching our heads on how to do that. And we reached out via our collaborators at Hopkins, specifically Tony Camerato, to Tom and Wacom. And we went to Chicago and we took some human muscle and we stuck it and we wanted to see if it could diffract. And I feel like we don't have to go over this again, but as a really, really brief overview, we're really interested in the equator. And so we're interested in the 1,0 intensity, which is from the thick filaments, and then the 1,1 intensity, which is from the thick and thin filaments. And as a very, very brief reminder, increases in 1,110 means that the thick filaments are activated or structurally on, whereas decreases in I110 means that the thick filaments are structurally off. And we'll be kind of referring to this paradigm throughout the remaining slides. And so here, I always like showing this. This is really neat. These are X-ray diffraction patterns from human patients. So on the left, you see a non-failing control, and on the right, you can see a half-ref pH patients. And correct me if I'm wrong, Wei-Kong, but to my knowledge, these are some of the first ever human diffraction patterns, at least from cardiac muscle, that have been obtained, which is really neat. This is frozen, by the way. This is all flash frozen cardiac muscle obtained at explant. So this is really, really exciting for us. And so what we did was we simply quantified the equators. And so I have a quick schematic and an example of a control diffraction pattern, specifically the equator here, and then an example of a patient with RV decompensation. And you can kind of see visually in this cartoon that the I1,110 ratio is off in these patients. And when we quantified it at PCA8 in non-failing controls versus the patients with RV decompensated RVs, we see that it is indeed really, really depressed. So what do the compensated RVs look like? Is this going to be like what we saw with the myosin isoform, where it's similar to the RV decompensated, or is it going to be similar to the controls? And when we did those patients, we actually found it to be really similar to the controls. And it's significantly, the equatorial intensity ratio is significantly higher in these compensated RVs versus the decompensated RVs. So what this suggests to us is that the number of functionally accessible heads, or at least the relative association between myosin and the thin filaments, is actually reduced in the RV decompensated, but not the RV compensated patients. And just to kind of provide a bit of a reference as to what this intensity ratio change means, we took some human control muscle treated with mavacamten, and you can actually see that the change with mavacamten is similar, if not slightly smaller, than the change we see in these patients at rest. Which again kind of highlights how uniquely depressed their contractile function must be. The next thing we decided to explore was calcium activation, and specifically calcium activation of the I110. So shown in this plot right here, the same three kind of scatter plots from the previous slide for the black non-failing controls for the RV compensated patients and the RV decompensated patients, which were again significantly lower than the compensated patients in the non-failing controls. And when we increased saturating calcium concentration at PCA5, we see as expected that the non-failing controls go up. And we actually see that the RV compensated patients also increase their equatorial intensity ratio. But to our surprise, we find that the relative change in I110 with calcium activation in the decompensated patients is quite reduced. And the P interaction between the group and calcium effect in decompensated versus compensated patients is actually significant at 0.01. So what this suggests to us is that calcium activated increases in the equatorial intensity ratio are reduced in RV decompensated patients, as at least compared to RV compensated patients. When we look at lattice spacing, just as a sanity check to see if that was any different, it really wasn't at all of the calciums and all of the in all of the groups. So as we kind of talked about, has been talked about quite at length throughout the entire last couple of days, there are lots of mechanisms by which I110 equatorial intensity ratio can be depressed, and we decided to explore one possibility, which is the proportion of myosin in the super relaxed or disordered relaxed state. Now, again, these aren't one-to-one, but myosin, disordered relaxed myosin has a higher ATPase activity, and in certain situations can correlate with a normal equatorial intensity ratio. And similarly, super relaxed myosin has a depressed ATPase activity. And again, in certain situations, it can correlate with the depressed equatorial intensity ratio. But this is not necessarily a one-to-one correlation, as demonstrated really nicely by Safi in his talk yesterday. So in order to do this, and given that the human samples are quite precious, I ended up building a single myocyte loaded MANT ATPase system in which I have actual control over sarcomere length. So I made sure I did all my measurements at the same sarcomere length as the mechanics. And then this is kind of just a cartoon average tracing from about 20 or 30 control myocytes. And here you can see the characteristic bi-exponential decay. We attribute the kind of first half of this, the fast phase, to predominantly disordered relaxed myosin, and the second half slow phase to super relaxed myosin. Again, that interpretation is actually somewhat limited, as we're starting to learn now, but This is the way we took it. And when we look at the myocytes from RV decompensated patients, we see that it's the ATPase activity is significantly slower. And that's shown here in red from about 20 or 30 myocytes from all these patients. Moreover, when we look at the blue RV compensated patients, we see that actually it seems to have some altered kinetics in this fast phase right here. And actually when we actually quantify the amount of DRX myosin in these three groups, we see that it's not significantly different between the RV compensated and non-failing controls, but the RV decompensated patients have reduced DRX myosin. So what this suggests to us is, at least in this particular disease situation, the proportion of DRX myosin seems to be explaining in part some of the results we saw of with the depressed equatorial intensity ratio in these patients. Just to kind of show what the kinetics of the slow and fast phase look like, you can see that there's not really any differences in T2, which is the time constant for the slow phase, but as shown by the cartoon, we did see a significant decrease in T1. This could be a form of compensation or a mechanism of compensation for these myocytes, but that's something that we're actively exploring. Now, up until this point, all of the data we've seen is basically a correlation, and we haven't really quite established whether or not the changes that we've observed in equatorial intensity ratio or percent DRX myosin actually correlate one-to-one with force production. And so we did this with two sets of experiments. The first, we took muscle or cardiomyocytes from both groups of patients, and we treated them with the myosin ATPase inhibitor mavacamten. And so our idea was that, in these RV compensated patients who have, elevated equatorial intensity ratio normal compared to controls, mavacamten should bring it down. And we expect to see, and we did observe, a decrease in the equatorial intensity ratio and a concomitant decrease in isometric tension. But in the patients with, already excess, super relaxed or too little disorder relaxed myosin, we didn't really see much of a response with mavacamten, as demonstrated on the right by the not significant differently change in equatorial intensity ratio and the not significantly different change in force, though there is a significant right shift that we observed. Similarly, we did the kind of converse experiment now with deoxy-ATP in these two groups of patients. So what we found was that deoxy-ATP didn't actually have too much of an effect in our RV compensated patients, we think, because they already have a normal amount of SRX myosin. But I will say that we are underpowered to make any claims there, just given how the machine learning group stratified out. We don't see a significant change in equatorial intensity ratio, though we see that most of them do go up. We don't really see a significant change in the force profile. But we did see that in deoxy-ATP was able to rescue the RV decompensated myocytes, and we see that not only did equatorial intensity ratio go up, but so too did isometric tension. So what this suggested to us is, while these myosin heads are off, the contractile machinery is present, and this could be a potential pharmacologic target for these patients. The next thing we decided to do was look to see whether or not there's a correlation between the amount of DRX myosin you have at rest and how much force you produce at 2.5 micromolar of calcium. So shown here are paired measurements from the same patients in which you have T2.5, which is the isometric tension at 2.5 micromolar of calcium on the y-axis, and the percentage of disordered relaxed state from the MANT ATP on the x-axis. And we took each of these patients and treated them with mavacamten, as shown, which are the squares, and then we fit all of that into a linear regression, and we see a weak to moderate correlation between isometric tension and DRX myosin, and that's significant with p equals 0.003, even after adjusting for patient group as a covariate. So this suggests to us that while the fact, and really the mavacamten experiments really helped solidify that for us, was that this is not simply just a difference in heart failure versus control, but this really seems to be a spectrum at which you can predict kind of how much force you produce in systolic conditions from diastolic or resting percent DRX myosin, which is pretty neat, we thought. So now that we've kind of established that there seems to be a relationship between the percentage of DRX myosin we have at rest, as well as the and isometric tension, we decided to look at the final behavior that we were interested in, which is the length tension relationship. And so to do this, what we did was we took the same skin myocytes and we performed the MANT ATP assay at two different sarcomere lengths. And again, these are paired experiments. So we took a myocyte, we stretched it to 2.1 micrometers, we acquired a MANT ATP fluorescence trace, and then we took that same myocyte, stretched it to 2.4 sarcomeres length, and then acquired a MANT ATP assay again. And then what we did was we quantified the percentage change or the relative change in disordered relaxed myosin. And Weikong showed this, at least the first part of this data, in non-failing control patients yesterday. And here you can see that we do see that we are able to recruit DRX myosin to stretch in control patients. But what was really interesting to us was that in the HFrEF patients, we see that this behavior seems to be reduced. And it's reduced across the board in all groups, and both compensated and decompensated patients. And we thought that this must be due to the difference. This may be contributing to the depressed systolic stiffness that we observed in our length tension relationship. But we don't really have enough evidence, at least with these sets of data, to prove that, given that these are taken at rest and those measurements are taken during calcium activation. And so to address this question, what we decided to do was we worked with collaborators at University of Kentucky, specifically the Campbell Lab, and we used their online FibroSim tool to kind of model what happened if we impose the two behaviors that we measured. So as a brief kind of overview on FibroSim, you can program in the proportion of SRX myosin, the proportion of DRX myosin, as well as the rates of transitions between them. And we effectively programmed two conditions in addition to the control or baseline condition. Condition one, we set this percentage of SRX myosin to 25%, and we prevented any SRX to DRX transitions. This is supposed to represent our, RB compensated patient group, where they have about a normal amount of SRX myosin, and they have reduced transition from SRX to DRX based at least on our stretch data. And then we have condition two, which has excess SRX myosin, in this case 50%, and again, the same idea of no SRX to DRX myosin transitions. And when we impose these two conditions in the model, we find that the results are actually quite similar to what we observed experimentally. So from left to right, we see the black controls, condition one in blue and condition two in red. We can see that the isometric tension relationship appears to be normal, but somewhat mildly depressed in condition one versus control, which is what we observed. And it's even more so depressed in condition two versus control. And we can see that the slope of the active stress versus sarcomere length relationship is depressed in both condition one and condition two, as is the power. So we think that this is a really interesting behavior, and we think that these two behaviors seem to at least be able to sufficiently explain a lot of the differences that we observed experimentally. And we're now kind of chasing potential physiologic and molecular explanations for these phenomena. So as a kind of a brief summary to go up until this point, we find that depressed calcium activated tension predicts RV failure, reflected by measures such as PAPI. Myocyte stiffness and maximum power output do not predict RV failure. And so this is something that we're really interested in to see why is it that our standard clinical indices are kind of failing to capture this really fundamental phenotype. Next, we find that cells with low calcium activated tension have inactivated thick filaments and reduced percent DRX myosin, and increasing this can actually augment cardiomyocyte force. Moreover, we see that depressed power and systolic stiffness associate with myosin isoform switching and reduced stretch-dependent recruitment of DRX myosin. And perhaps we think that pharmacologically recruiting SRX myosin into the DRX conformation in patients with, for example, reduced PAPI may help to treat RV failure in patients with HFrEF PH. And so this was really exciting, and I'm really thankful to all the work and all our collaborations with everyone to get this work started. And now we're kind of moving on, and I wanted to, as we have a bit of time, I wanted to move on to what the next chapter of this project is looking at, specifically in patients with HFpEF. So unlike patients with—
Hey, Vivek, can I ask a question before you move on?
Sure. Yeah.
Did you mention what kind of pulmonary hypertension this is?
Yeah, they're all group 2. So I did not mention that, but they all have wedge greater than or equal to 15. They all have, of course, mean PA pressure greater than 10 or 20. And actually, they're all transplant, so their PVRs were pretty low. They're all less than 3. So none of them were combined pre-post capillary.
Thank you.
Yeah, and then of course, Steve, as you know, has done a lot of work in group 1 PAH and systemic sclerosis PAH, and we can chat about that later if we want. But yeah, now I'm kind of going to transition to, well, this is heart failure with reduced ejection fraction. What about heart failure with preserved ejection fraction? And so I'm not sure how many in the audience are familiar with this syndrome, but I thought I'd spend a few minutes going over it in brief. So HFpEF is really a clinical diagnosis. And here is the definition. It's an LV ejection fraction on echo greater than or equal to 50%. and signs and symptoms of heart failure. So these would be like shortness of breath, a symptom called orthopnea, which is when the patient lies down because they have dysregulated fluid homeostasis. They get short of breath when they lie down. And then some evidence of a cardiac problem. This can be anything from elevated natriuretic peptides to left atrial enlargement and left ventricular hypertrophy, as shown by the arrow on the right, diastolic dysfunction, and elevated filling pressures. And the definition of HFpEF is necessarily broad, and as such, there's actually a large amount of heterogeneity within HFpEF patients, and that makes it incredibly difficult to study. So of all the phenotypes that I've shown here, the classic one that they teach you in medical school, for example, was that HFpEF is thought to be a disease of predominant left ventricular hypertrophy and hypertension with impaired relaxation and diastolic dysfunction. And so, people looked at this was kind of the HFpEF of the 90s and the early 2000s. And Walter Paulus's group did a lot of really important work kind of elucidating the cardiomyocyte phenotype in these HFpEF patients. And so this is one such a summary of one paper from the group. And basically what they found is that, these patients have their myocytes derived from these patients with HFpEF. And this is, again, specifically a diastolic heart failure phenotype of HFpEF. They have elevated passive tension at low PCAs and actually normal, not significantly different changes in tension at higher PCAs. And of course, passive tension is higher in HFpEF versus controls. But this is not the HFpEF that we see. Obesity has really, really changed the phenotype of HFpEF. So before, a lot of patients with HFpEF had significant LVH and diastolic dysfunction and hypertension. But now, even at Hopkins, the median BMI of our HFpEF cohort is 38, which is very, very high. And this kind of comorbid obesity has resulted in HFpEF kind of evolving as this multi-system syndrome that has both pulmonary, liver involvement, renal involvement, and the adipose tissue itself has some sort of role here. And we don't really know what's going on, but what we do know is that the patients with obesity and HFpEF, they have reduced exercise capacity and they have worsened quality of life. And this is even more so than those patients at the same BMI without HFpEF. And importantly, these HFpEF patients are doing worse overall. So this is a survival curve for all-cause mortality in patients with obese HFpEF in red versus non-obese HFpEF in blue. And you can see that at 5 years, the survival is probably 75% in these patients versus like almost 80% or 85%, and this is significantly different. So what the question that went to our minds is, what do the myocytes from these obese HFpEF patients look like? Do they look like the myocytes that Walter Paulus has shown previously, or do they look something completely different? So first, I'm going to kind of show you the non-filling forced calcium and non-filling length tension relationship here. This is now passive as opposed to active. And then in purple, I have the HFrEF PH patients with the RV dysfunction that we've been talking about previously. So this is kind of our control for what contractile dysfunction is supposed to look like. So let's first go ahead and look at patients. So we took a cohort of 46 patients and stratified them into one of three groups. The first group is patients with LV hypertrophy and hypertension, which we're going to call HFpEF HHD. And these patients are very similar to what Walter Paulus had published on previously. They're not very obese, though their median BMI is still about 30, so it's not normal for sure. And then we looked at the patients with significant obesity with the median BMI greater than 40. And then finally, we looked at a subgroup of patients with both left ventricular hypertrophy and obesity. So let's look at what their myocytes look like. So this is the HH patients. And this is exactly what was shown previously. You see that there's normal force at systolic calcium concentrations, and you can see that passive tension is increased. But what do the obese patients look like? This is the group that is now kind of the predominant phenotype of HFpEF that we observe. These patients have depressed force, and this was very, very shocking to us. We were not expecting this at all. Yes, their myocytes are slightly stiffer, though this did not reach significance compared to controls, but we do see that their force is as low as it is in heart failure with reduced ejection fraction. Now remember, these patients have a normal ejection fraction. They don't have a reduced ejection fraction. So first, ejection fraction doesn't really tell us about what's going on. And second, it is not true that HFpEF is a disease of predominant diastolic dysfunction anymore. here you're clearly seeing systolic dysfunction. And there have been several studies by Barry Borlaug and others that have shown reduced contractile indices on more sophisticated testing in these patients. Now, what about the mixed patients? They have both LV hypertrophy as the red group, they're matched, and the same BMI as the blue group. Do they look like the red group, or they look like the blue group, or are they in the middle? And they look exactly like the blue group. So what this tells us is that obesity seems to be doing something to the myocytes, and that's causing their forces to be very, very low. And that has really, really changed what's going on. To hammer this point home, here's the correlation between Tmax and BMI. And it's very, very tightly and strongly correlated in HFpEF. And so we think that obesity is a predominant driver for this phenotype. And basically, the second half of my thesis project is really exploring what's going on here. But the first thing we decided to do was X-ray diffraction, because that has shown some very interesting phenotypes for at least for HFrEF in the past. And so we did X-ray diffraction this time from HFpEF biopsies. And Wei-Kong and Axel, I know who are both in the call, can tell you that this is very, very technically challenging from these really, really small biopsy species. So again, would like to thank them for all the hard work for compiling this data. But shown here is the non-filling controls. And again, here's a reference depressed equatorial intensity HFrEF with RB dysfunction group. So the hypertensive hypertrophic red group, again, who had normal force before, they showed equatorial intensity ratio actually similar to controls. But what about the obese group? Do they look the same or do they look like HFrEF? And to our surprise, they looked exactly like HFrEF. The equatorial intensity ratio is significantly depressed in these obese patients. And it's significantly depressed compared to the hypertensive patients as well. So again, this seems to be an obesity thing, and obesity is doing something that we don't quite understand. So now we're kind of looking at what the implications of these findings are, as well as what patients with both of these phenotypes look like, and that data is upcoming. So in summary, the obesity pandemic has really, really shifted the phenotype of HFpEF. Myocytes from HFpEF patients show reduced Fmax, but not myocytes from LV hypertrophy and patients with systolic hypertension. And the equatorial intensity ratio in skin muscle from obese HFpEF patients is reduced as much as what we see in HFrEF patients. And so I'd like just to thank everyone from our lab. I'd like to thank Wei-Kong and Tom and Axel and everyone who's helped me out with this project. And I'm excited to kind of keep moving forward. And I'm happy to take any questions now. Thank you.
As usual, great talk, Vivek.
Thanks.
Okay, if anyone has questions, go ahead. You can unmute yourself, ask the speaker.
Hi, Vivek. I guess I put my question in chat, but do you know anything about fibrosis in these patients?
Yeah, so actually, this is a paper that came out from Virginia Hahn and our group a couple years ago in Jack heart failure. And it turns out that the fibrosis in HFpEF patients, if you age match it, is not significantly different from age match controls. So this is not a fibrosis thing.
Thank you.
Now, Walter Paulus has shown that the titin is significantly stiffer in these patients, and this seems to be PKG dependent. So that's something that we're exploring, of course, in the HH patients.
Hi, Vivek. Great talk. I might have missed this, but in the beginning of your talk, you mentioned a machine learning model. So what kind of model was that?
Yeah. Yeah, it's a good question. I glossed over it because it's a bit math heavy, but I did something called a Dirichlet process mixture model. So what that the classic example, it's called the stick breaking procedure. So imagine you have an infinitely long stick, and then you break it along the length of the stick with some probability. That's a cluster. And then you keep breaking the stick until you completely explain the variance in your data. And so the advantage to that model is twofold. First, unlike most unsupervised machine learning models, it actually learns the cluster number. So the cluster, in most machine learning models, the clusters are actually an input that the user has to put in, but here it's a random variable, and actually learned that. So 2 clusters is actually, not only is it it's not something that I decided, it's something that the model itself decided. The other model we used was a simple just k-means clustering, and that was for the clinical data. Yeah.
Thank you for the explanation.
Can I just go back to something you mentioned quite early on in your SRX DRX measurements? You said that the DRX T1 was much smaller in your control samples than the normal. I forget which samples it was, but there's a big difference in the DRX T1.
Yeah,
among
it was
those
in the
patients.
blue RV compensated patients. So the patients with heart failure, but with normal forced.
And that was a very, very big change, as I seem to recall. It was a big change in that, which seems to me really quite significant as to why that phase is so much faster in those patients. If you looked at that, any more detail?
Yeah, it's a good thought. I actually don't. No, I saw that too, and I agree. So we haven't really looked into it yet, but we're kind of doing, fishing expedition experiments to see perhaps if there are changes in phosphorylation or something like that that could be associated, and so that's something we're actively pursuing.
Yeah. Okay, thanks.
Yeah, thank you.